All right, we now have everything loaded into Benchling. So we've got 3 DNA sequences and 1, 2, 3, 4, 5 primer sequences. I know we're kind of going through the steps and you don't totally understand why you're doing what you're doing yet, but we'll get there, I promise. All right, so the next step is to attach the primers to the DNA sequences where they match. And this is a really powerful thing that the computer is able to do for you, right? So we're going to take, it's essentially doing that needle in a haystack search very quickly, right? And it's going to say, these primers will stick and it'll show you where. So to do this attaching, you're going to come to your right sidebar, and there's this little symbol here that looks like primers. Underneath the scissors, those 2 little arrows. So we're going to click that, and it says, you don't have any primers yet. And you're like, that's correct. We want to attach existing primers because we already uploaded all of our primers. So we'll choose attach existing. All right, so there's lots of settings in here. Find binding sites for oligos in specified folders. The folder that it's specifying is example project. We know we're within example project because I can see that in the top left corner, and our bio 288 folder is within there. So that means it's going to search this folder and see our primers here. So that's good. All right, how many matching bases do we want? I think the default is 14. I'm leaving it at that. Separated by whatever number of mismatches. So this is telling you, you're telling the computer, this is how many wrong bases in a row you can have, and you're still going to show me that result. And we're going to leave that on 1, which I think is the default. And then the next one, and no more than blank total mismatches, is the total number of incorrect bases that you can have. And we're going to leave that on 2. I think it's defaults to maybe 1 or 0. I don't remember, but set that to 2. I know I told you that primers will only stick if the sequence matches absolutely perfectly, but that isn't entirely true. It comes down to your annealing temperature, right? So the temperature that you set the thermocycler to be when the primer is trying to stick will change how lax or how strict that interaction is. And we want to allow 2 mismatches, and you'll see why in a little bit. And then this is the melting temperature, and you can put a range here. I think it defaults to 48 to 100, and I'm not going to mess with that. I'm not going to check where the entire primer matches because, again, I want to allow those couple of mismatches. So we're going to leave those unchecked. And then I'm not going to narrow the range within my sequence that it's searching. I want it to search the whole thing. And now we're going to click Find Binding Sites. And in about a second, it's going to say, here's where your primers bind. All right, okay. I'm going to move my zoom menu that you can't see. All right, so it found a matching location in our sequence for primers 23, 24, and 25. This is the number of the bases of where it binds and which strand, positive being one, negative being the other strand, the melting temperature, and then here it's showing the bases. Everything highlighted in blue means that it matched perfectly. And we want to select all 3 of these, and because we want to attach them all, and then the attach button is way up here in the top right. Attach selected primers. So we're going to choose that. They turned green now, and it grayed out the checkbox indicating they're attached. And then in my menu here in the top left, I'm going to go back to my sequence map. And now here are my primers. Right? So here is primer 23. You can see it's binding to the top strand, and this arrow indicates the direction that it's facing, pointing this way. So meaning that this is the 3' end of the primer, and DNA is going to get added over here. It's going to grow in this direction. Here's primer 24. You can see it's annealing with the bottom strand and pointing up. So this is what we refer to as a reverse primer. This is pointing upwards. And then here's 25 down here. We can see it. And then if we scroll, right, there aren't any more. Those are the three that it found on the sequence. So go through that exact same process with your other two DNA sequences. So go to your Abramanii sequence, attach primers with the same settings, and your Staph epi sequence and attach primers with those exact same settings. And I will see you when you're done.